you should note, part of this is just got to chalk it up to memorization. There's no way around it, and a lot of it. So don't think, I don't really need to know that. I'm just going to let you know you need to know a fair amount of stuff. Now, there's some things you definitely don't need to know. Like, do you need to know every single little structure that you've gone through? Probably not, but you probably do need to know a few of them, like glucose, fructose, and some of the ones involving some of the major regulatory steps and stuff like that that you spend a lot of time on. So if Dr. Leffler focused and spent a fair amount of time on stuff, then that's where you need to focus and spend a lot of time as well. In your studying, you're definitely going to want to go through all of his slides. So when you go back through his slides after this weekend, or after tonight anyways, if there's anything you don't understand, then you better make sure you understand it. And then when you take the practice test, same thing. If there's anything you couldn't answer, you need to go back and figure out why not. So there's a couple things. Memorizing everything will probably get you a B on this test. Memorizing everything. Did you hear that? Scary, huh? So here's the deal. Memorizing everything is not enough. You need to organize it in your head as well. So, and how you organize that, so I'm going to try and hopefully help you with that a little bit throughout the night and just how I present it. But how it is organized in your head is also related to not just memorizing it, but then organizing it, but then also understanding how to apply it to maybe something a little new. You know, say we added this inhibitor and it stopped this step in glycolysis, what would you expect? You know, that kind of a thing. So memorizing everything is the first step you need to take to getting to that point. So, but it's a step beyond. Cool. So everything you guys are covering deals with metabolism. So, and part of that is catabolism. What is catabolism? So breakdown of what? Cool. So, and presumably for what purpose? Yeah, make energy, right? So, and that's what we're going to start with here. We're just going to get the big picture on catabolism here. So, and for pretty much every organism, they start with glycolysis. So glycolysis, what's your major substrate that we typically talk about, spend the most time with? Glucose. Sweet. So here glucose goes into glycolysis. So what comes out of glycolysis? Pyruvate. Notice glucose has 6 carbons. Pyruvate's got 3, so you get 2 pyruvates. So, and then what you do with pyruvate depends on if, you know, what kind of organism or what kind of cell you are. If you're certain anaerobic microorganisms, or if you're red blood cell, then you got to kind of go the anaerobic pathway, and you're not going to get a big energy output out of this, right? So, but if you're aerobic, where are you going to take that pyruvate? So you're going to send it off to the citric acid cycle, converting it into acetyl-CoA on the way there. Once it goes to the citric acid cycle, so what do all the carbons of glucose ultimately, what's their ultimate fate after the citric acid cycle? It's what you're exhaling right now. It's your carbon dioxide. Notice you can kind of follow the carbons in blue. 6 carbons, 2 3-carbon chains. There go a couple carbon dioxides. You got 2 2-carbon acetyl-CoAs left, both of which net result get turned into carbon dioxide. So all the carbons in glucose end up, their fate is carbon dioxide. So we'll see that along the way, if you're going the aerobic route, one of the big goals is to produce lots and lots of reduced cofactors, NADH, FADH2. What do we do with those? So we'll send them off to the electron transport chain, and we'll deal with that next test. But the electron transport chain ultimately is going to turn that into a proton gradient and use that to drive ATP synthase and make boatloads of ATP. About how much, we don't have to go too specific on this, but about how much ATP can you get for every glucose molecule? So right now about 30, but depending on who you talk to, some say 32, some will say as high as almost 40, depending on what textbook or what professor you go with. Right now, suffice it to say, let's just round it to around 30-ish. We'll get a little more specific in the next section with electron transport for next exam, but not for now. So the big energy payout, so in cellular respiration, we'll see when we finally deal with the electron transport chain. So, but suffice it to say, we're going to deal with glycolysis. We're going to look at anaerobic. We're going to look at aerobic all the way up through the CAC cycle, sometimes also called the TCA cycle or the Krebs cycle. If I call it the TCA cycle, the tricarboxylic acid cycle, I'm sorry, that's the way it was taught to me back in the day, and I'll probably call it that much of the night, forgetting that you guys may not have seen that, but it's the same thing. CAC cycle, TCA cycle, Krebs cycle, same diff. Cool. Any questions before we start breaking this down? Just want to make sure you see this big picture. We're making some energy. So we're going to make a little bit in glycolysis. We'll make a little bit more in the citric acid cycle. So But end of the citric acid cycle, we got lots of NADH and FADH2 that will shuttle off the electron transport chain. And when we study that later, that's where you'll see the big energy payoff. So glycolysis, good times, right? So is glucose the only substrate for glycolysis? So it is not. We can shuttle fructose or galactose in there and stuff like that. So we do not have time to cover everything tonight. That's one of the things I'm going to leave to you. So there's a couple of short slides about how those get introduced into glycolysis. I'm going to leave that on you. We're just going to talk about glucose and focus there. Spend a whole lot more time there. Whole goal here is to turn glucose into pyruvate. If you keep track of how many carbons so your intermediates have, it's often a little bit easier to keep track of where you're at in the process. So glucose has how many carbons? 6. Sweet, and here's glucose. Glucose, aldose or ketose? Aldose, it's an aldehyde sugar. So, and you definitely, this is one structure. One, you should have known it for last test, but you gotta definitely know this one. No way around this. So your hydroxyl groups, if you follow them on the Fischer projection, right, left, right, right. So one thing to note, I'm gonna follow glycolysis with the Fischer projections. So rather than viewing it through the cyclic Haworth projections, which lots of students find confusing and for good reason, I'm going to follow it through the Fischer projections. It might make it just a little bit easier on you. So you don't have to know every structure we do, but definitely you want to know some of the major ones, especially in some of the major steps that you spent a lot of time in lecture on. So, but we'll start with glucose. So we'll end up at the very end with a couple of pyruvate molecules. So how we get there and what's important? Good times. So what do we first turn glucose into? Glucose 6-phosphate. So how does that differ than plain old glucose? Good. Carbon 6 way down here at the bottom. Is now going to end up phosphorylated. The rest of it will pretty much stay the same. Let's get this correct here. Cool. From here on out, rather than drawing out phosphate every time, so there's your phosphate group. I'm just going to call that a giant P. So for simplicity's sake. So wherever we have a phosphate attached, I'm just going to call it a P. Notice glucose, carbon 1, 2, 3, 4, 5, 6. So glucose 6-phosphate. There it is. On carbon 6, its alcohol has been converted into a phosphoester here, or a phosphate ester, same diff. So but from here on out, I'm just going to put a P in a circle where we phosphorylate it. Cool. What enzyme does this? Hexokinase.
When you draw the phosphate group, you always want 2 negatives on there?
So yeah, 2 negatives on the phosphate. So hexokinase. So first part that's important here is ACE. When you see ACE as a suffix, what does it always mean? Enzyme. So next part is we're going to get to know a few others. What does kinase actually mean? It phosphorylates it, and what does it use to do the phosphorylating? Yeah, ATP hydrolysis usually to do some sort of phosphorylation. Where things get a little tricky is a lot of enzymes do reversible reactions. They catalyze both directions. And so sometimes you see something named a kinase, and you're like, why did they call that a kinase? Well, they might have called it that for the reverse reaction. So just keep that in mind. So but in this case, hexokinase, kinases phosphorylate things usually, the context we study them, and they usually use ATP to do this. And so in this case, we are indeed going to convert ATP to ADP. So one thing to note, I'm not going to balance these out. I'm not going to show you, yes, we added water to ATP, and a product was also inorganic phosphate. I'm just going to use this simple system of nomenclature or symbology, if you will, ATP to ADP. So in this case, a kinase, you're usually going to see ATP hydrolysis coming along with this. So this is really important. Where does the cell get glucose from? What's that? Lots of stuff. It might come from gluconeogenesis, but assuming, so it's coming from the diet. So where's it coming from then? So coming from your carbohydrates, but where's the cell get it from most immediately? So from your bloodstream. So coming from the bloodstream to the cells. So the glucose is going to get transported into the cell. So it comes into the cell as glucose. If we don't phosphorylate it, that glucose might leave. So following the concentration gradient to exit the cell back out. But by turning it into glucose 6-phosphate, is it glucose anymore? Plain glucose? No. And so all of a sudden we've changed the concentration gradient. There is no glucose 6-phosphate outside the cell in the bloodstream. It's just in the cell. And so in this case, being charged, I don't know, actually that probably is not important, but being charged, charge things across the cell membrane back into the bloodstream? No, don't have to worry about it going back out. So, but here, getting it into the cell immediately gets phosphorylated. That way we keep it in the cell. Keep it charged, keep it from crossing the membrane. Cool. So our goal is to make energy. How are we doing? Not so good yet. Sweet. So what do we convert this into next? Fructose 6-phosphate. So if you look at your glucose, so fructose looks just like glucose. It's an isomer of glucose. Only difference is fructose is not an aldose. What is it? It's a ketose. So we're going to have an enol tautomerization going on here, again catalyzed by an enzyme, but you're going to end up with your carbonyl at carbon 2 and the alcohol up at carbon 1. But everything else stays exactly the same. This is your fructose 6-phosphate. Nothing else is different. So if you know glucose, knowing fructose is pretty darn easy. So all we've done is isomerization here. So, and what's our lovely enzyme that does this isomerization? There are a couple of different names. So glucose phosphate isomerase. Why in the world would they ever call it that, right? So here we've got glucose 6-phosphate isomerizing, hence the name. So oftentimes understanding what's going on also makes it a lot easier for memorizing some of these names. A couple places it may not be so clear, but we'll make it clear along the way. Cool. So all we've done, isomerize this. Where do we go from here? Add another phosphate group. Where, well actually, what's the name of what we're ending up with at this next step? Cool, so if we've got fructose 6-phosphate here, we're going to end up with fructose 1,6-bisphosphate. Guess where we're putting a phosphate? Right there, but nothing else changes. Cool. What did we use to get there? Really important enzyme. What's this guy? PFK1, what does that stand for? Phosphofructo what? Kinase. Oh, sweet. It's a kinase. What do kinases do? Phosphorylate things. Sweet. That's what we did. We phosphorylated things. And what do they use to phosphorylate things typically? ATP. So this is another step. Which is going to involve ATP hydrolysis. So what's the goal of glycolysis again? Create energy, and usually what form does that energy typically take, at least initially? ATP is usually kind of our energy currency for the cell. So how are we doing? We're still not doing good. Up till now, all we've done is use up ATP, but that's going to come to a quick end. So the two steps we've had, both with hexokinase at the beginning and now with PFK1, we've used up 2 molecules of ATP. This is the energy investment. You got to invest before you can get a return on investment. We're about to start getting our returns, though. So, but again, another kinase, another molecule of ATP used up. So, and we now come to our first high, I guess he's not really a high energy emitter yet. We'll see that very shortly. Cool. How many carbons we got still? 6. That's about to change as well.